Hello everyone. Thank you for coming today for today's presentation. My name is Axel Villagomez and today I'm going to talk about our natural products and options for cancer treatment. This title is based on the anti-cancer activities of Vernonia amygdalina, lives on 41 breast cancer cells through PI3K pathway. I want to start talking about key aspects of breast cancer. As you may know, it's one of the main causes of death worldwide. And this disease has many factors that influence the risk of developing breast cancer, such as reproductive history, long exposure to hormone therapy, dietary factors, radiation during breast development, and also breast cancer related congenital genetic factors. And also, this disease is characterized by an uncontrollable growth of the breast cells, and the most common type starts in the milk ducts or glands. One of the key pathways that is part of the breast cancer is the phosphoinositide 3 kinase or PI3K pathway, which regulates the cell growth and proliferation. Many authors have described that this pathway is often dysregulated in cancer, and there can be many causes such as mutations, amplifications, deletions, methylations, and post-translational modifications. But also, other authors have described that this pathway is vital for apoptosis, malignant transformation, tumor progression, and radioresistance. So, throughout the years, some other researchers have described also negative regulators and inhibitors that modulate this pathway. Because, as I mentioned previously, this is related to survival, and this pathway is dysregulated in cancer. It is really important to find a way to modulate this pathway, especially in cancer cells. So naturally, we have this phosphatase and tensine homolog, which is also called as PTEN, which is a negative regulator of this pathway. But also, there are many inhibitors developed throughout the years targeting one signal node or even two signal nodes at the same time. As you can see here, we have this PI3K protein and mTOR, which are the key points of this pathway. And there are many molecules that can act on both or only on the PI3K protein, and therefore affect the survival of the cells. So, having this in mind, the researchers wanted to know if this plant, which is known as Vernonia amygdalina, contains some compounds that can act directly on this pathway. This plant is comes from West Africa, so it is found also in some parts of America, especially South America. But previous research has described that there are many compounds such as flavonoids, sesquiterpenes, lactones, fatty acids, and steroidal saponins that have some properties or pharmacological properties such as anti-malaria, anti-inflammation, anti-tumor, or even anti-obesity. So the aim of this study was to evaluate the anti-cancer activities of the leaves extract of this plant on 41 breast cancer cells through the PI3K pathway. The authors kept this in mind because they wanted to see if there are new potential molecules that can be used as new therapies for breast cancer. So first, to achieve that, they took the plant and they used the air-dry powder leaves, and they fractionated with three solvents, N-hexane, ethyl acetate, and ethanol. The separation of compounds happened due to the differences on polarity, and then using the ethyl acetate fraction, they performed a phytochemical analysis using liquid chromatography and mass spectra, and the results were analyzed using this software. Once they obtained these three fractions, they performed the cytotoxicity analysis using the 41 cells. These were the conditions for the culture, and they inoculated this concentration of cells. So after 24 hours of incubation, they discarded the medium and they added the treatment with the fractions, and they used doxorubicin as control. After 24 hours, they added this MTT reagent, which viable cells convert into into purple formation crystals, which is an indicator of how many cells are still alive or viable. And then 4 hours after they stopped the reaction with 10% SDS, they incubated an additional 24 hours without light, and then they measured the absorbance at 595 nanometers, and the data obtained was converted to percentage of viable cells. Then they performed some analysis using flow cytometry. So for that, they focused on three aspects. First, cell cycle analysis, then an expression analysis of these two proteins, which, as I mentioned previously, are related to survival of cells, and also an apoptosis analysis. So first, with the 41 cells, they used the same conditions for growth, and then they inoculated this concentration of cells. Then they added the treatment with the ethyl acetate fraction, and after 24 hours of incubation, they collected the floating adherent cells into another conical tube. They washed and they separated the supernatant, and then with the sediment, they performed further analysis. For the cell cycle analysis, they fixed these cells using these conditions, and after washing and resuspension, they centrifugated and then added the PI reagent, which is used for staining DNA, and it's very useful for this fluorescent imaging. And after incubation of 30 minutes, they performed their analysis with the FACScan flow cytometer, which is a fluorescence activated cell sorting system. And depending on the DNA content, the percentage of cells in each stage of the cell cycle was calculated using this software. So the cell cycle, as you may know, it's G1, S, G2, and M. And using these same cells, then they performed the apoptosis analysis with the annexin V reagent, which was added to the sediment, and also they incubated an additional 30 minutes, and they performed the analysis with the same equipment. On the other side, they performed the expression analysis of the PI3K and mTOR, which are key nodes on the pathway. And they also fixed their cells using these conditions, and they performed this test using these antibodies against PI3K and mTOR and their respective signaling or label. And they incubated 10 additional minutes, and then they perform the same analysis using the FACScan equipment. So for the results, once they obtained the ethyl acetate fraction, they performed this phytochemical analysis to predict which compounds were present in this extract. So the results show that the ethyl acetate fraction contains diterpene, phenolics, flavonoids such as luteolin, diosmetine, and coumarins such as 7-hydroxycoumarin. So these are really important because they have some properties that are going to be discussed later on this paper. But at least we have an idea of which molecules are predicted to be present in the ethyl acetate fraction. For the inhibitory concentration, 50% of the fractions, they took these three ones and they tested the MTT. They performed the MTT assay on the 41 cells, and they observed that compared to the NXN fraction and the ethanol fraction, the ethyl acetate fraction was the one that presented the lowest concentration for the inhibitory, the IC50. So this means this was the fraction that produced the most cytotoxicity out of the three fractions they obtained from the plant. So their conclusion here was the cytotoxicity of the natural products is related to the content of active compounds in some plants, which in this case we can see that are present in the AF fraction. Once they performed the analysis with the cell cycle and apoptosis, they observed that there was an increase of cell death by modulation of the cell cycle. So in this case, we have in this area the control, and in the B area we have the treatment with a concentration of 12.5 micrograms per milliliter, which was around half of the IC50, to see if they have any effect on the cell cycle and also apoptosis. So first they observed that there was we have this histogram, and they observed that the content of DNA observed was accumulating more at the G2M phase, which was 34.22% compared to 17% at the control cells. So this part of the cell cycle, just for visual reference, it's when the preparation for mitosis and cell division occurs. So as you may know, we are expecting only a decrease, a plateau, and then a decrease of the DNA content observed in these stages, at this stage. But in comparison with the treatment, it's accumulating a little bit more compared to the control. So according to their analysis of the apoptosis, they when they added the annexin V reagent, we have these four parts of the plot which describe the type of or the stage which cells are going through, or which cells are going through. So in this case, this first part is viable cells. The second one are necrotic cells. These are late apoptotic or necrotic cells, and here we have early apoptotic cells. So again, we have in A we have the control, and B we have the treatment with the same concentration. And they observed that the percentage of control cells and cells treated with the EAF was 0.5% here and 3.13% in early apoptosis. And for the late apoptotic and necrotic cells, for control it was 0.52% compared to 5.12% with the treatment. And for necrotic cells, they observe here 2.99% compared to 22.75%, which indicates that the treatment at this concentration is causing not only necrotic cells or certain cytotoxicity, but also there's a distribution of how cells are dying, if they are at an early apoptotic stage or also at a late apoptotic or necrotic cells. Okay, so this they were also wondering if this cytotoxicity of the collection of compounds happened due to the modulation of the PI3K or mTOR expression. So in this case, they used again the same flow cytometry method. And as you can see, we have the histogram, which also shows the control and the treatment. And we have the evaluation of the PI3K expression and mTOR expression. So in this case, the treatment of EAF caused a cell accumulation in this M2 area, which represents the 7.59% compared to 4.27% of the control cells. So in this case, what it means is that when we have a greater area in this histogram, that it's low counts of this gene, it means that there are more cells that are expressing less protein compared to the control. So the effect of the ethyl acetate fraction is that there are less there are more cells that are expressing less amount of the protein, which also happens with mTOR. in this case, the difference observed was 9.22% versus 1.04 in the control cells. So at the same, we have the treatment and the control, and as you can see, there is a bigger amount of cells here that express low concentrations or less counts of the protein mTOR. Okay. So their analysis, they determined, some of the compounds found in Vernonia amygdalina, and they have been previously reported as some active molecules. So here, for example, we have some flavonoids and diterpenes and phenols, which, for example, one of them is apigenin, which is a flavonoid that has been previously described in other papers with anti-cancer activity when it's combined with this molecule, the 5-fluoroacyl and cisplatin. And the results describe that inhibits growth of these other cancer cells that are related to carcinoma. And they also describe that induces cell cycle arrest in the G2M phase. Another molecule is luteolin, which is a flavonoid that is known for its chemopreventive and cytotoxic agent, and increases the expression of these P53 and P21 in these A549 cells. And it has this anti-proliferative activity also in this U251mg and U87mg cells. And also, some of the coumarins, they also have described that inhibit the proliferation of cancer cells through cell cycle arrest. So their conclusion here is that the EAF has increased the cell's likelihood of undergoing apoptosis, both early apoptosis and late apoptosis compared with the control cells. So, for example, we also have some polyphenols, such as camphorol, quercetin, or ellagic acid that also can inhibit the cell cycle progression or induce apoptosis on human breast cancer. So they only play a little bit with the information that it's already available on the papers, on journals. And for example, they also describe the apigenin releases cytochrome C, which activates the caspase cascade, which is really important for then regulating the anti-apoptosis proteins such as BCL-XL, MCL1, but also upregulates these BAX, BAD, and BAK in T24 cells. So one of the key aspects is that they are only working with a collection of compounds, but it is very difficult to understand which compounds are responsible for which activities. So I think, as a critical opinion, I think that further studies are needed to elucidate the structure, the actual structure, and the mechanism of action of these predicted compounds from this ethyl acetate fraction. So, because they purificated and isolated these compounds only using fractions, there is no way to know which compounds are actually responsible for such activity. So first, I would like to suggest if they can perform further purification and isolation using open column chromatography, using silica, or maybe they can also perform chromatography using size exclusion, such as Cefadex. And once they obtain these compounds purified, then they have to identify, and for that they use this liquid chromatography and mass spectra technique, but I think it's not enough because they only took the information and they only predicted the types of compounds that they were present in the extract based on a compound library. But maybe some of these compounds can have some small modifications on their structure that can maybe enhance the activity or reduce the activity, or they can be they can be combined, so maybe they can be some kind of interaction between them that enhances also the activity. So for that, I think it's required to perform this analysis using also the most powerful spectroscopy techniques, such as NMR, that can be proton NMR, carbon 13 NMR, and also perform this uh, analysis to know or to elucidate better the structure of the molecule. I also think they require more cell assays, because they only evaluated the expression of two proteins, or yeah, the presence of these two proteins. So I think it's necessary to evaluate the anti-cancer activity based on the compounds found, not only on the collection. And once they perform this analysis, maybe they can focus on the pathway, but also maybe they can perform other parts of the path. Maybe they can focus on the research on other parts of the pathway, not only PI3K and mTOR, but using this same study design. I think they need to also evaluate the isolated and identified compounds as potential inhibitors of the PI3K pathway. So in this case, I think it's really important to continue the results, which are very promising. And also, I think it's another way to continue the research of novel compounds that are useful for cancer treatment. So with that said, I think we're done. Thank you so much for attending this presentation, and have a great day.